In this talk, we're going to look at how life originated on Earth. A simple definition of living organisms is that they are entities that are made up of organized structures called as cells. These cells utilize energy in order to grow, reproduce, and respond to their environments. Thus, any entity that is able to satisfy these conditions is a living organism. When we look at the history of Earth, then somewhere around 4 billion years ago, the oceans were formed. These oceans then allowed the formation of the first living organisms, which is estimated to have arose around 3.5 to 4 billions of years ago. For the first cell to form, many different events had to have occurred. In order to form living cells, we first need to form the building blocks that are present in living organisms. These building blocks are usually polymers of simple organic molecules like amino acids, purines, pyrimidines, or fatty acids. In order to explain how these simple organic molecules were formed on Earth, there have been different ideas and hypotheses that have been proposed. One idea is that these simple organic molecules could have come to Earth from asteroids or meteorites that landed on Earth. By analyzing different asteroids and meteorites that are present on Earth, scientists have found different types of organic molecules present in them. Another hypothesis to explain the formation of simple organic molecules is the idea that they were synthesized at deep sea vents. Deep sea vents have higher temperatures and pressures, and when scientists did experiments that simulated the composition of the oceans present on primitive Earth, and when they put the pressures and temperatures that are normally observed at deep sea vents, they were able to see the formation of simple organic molecules in those types of systems. Thus, this hypothesis has also been supported by different experiments done by scientists. Another hypothesis is that these simple organic molecules could have been formed from compounds that were present in the atmosphere of primitive Earth. In a one-of-a-kind experiment for their time, the scientists Stanley Miller and Harold Urey performed an experiment where they took a mixture of gases that mimicked the primitive atmosphere of Earth and provided periodic electrical sparks which simulated lightning. They then analyzed the water, which was mimicking the primordial oceans of primitive Earth. And when they analyzed the water, they were able to find the presence of many different amino acids, including other organic compounds. Thus, they were able to show that with the presence of simple gases like water, methane, ammonia, and hydrogen, they were able to form different types of molecules that are present in living systems. Once simple organic molecules were formed, these molecules should have the ability to assemble to form complex macromolecules commonly found in living systems, like nucleic acids, polypeptides, so on and so forth. Scientists hypothesized that sand, rock, clay, or mineral surfaces that were present on primitive Earth allowed the formation of these polymeric substances. To support this hypothesis, an experiment was done where ribonucleotides were put into a system where there was some clay mineral. And what they found was after some time, they were able to detect the presence of intact ribonucleic acids, which are the polymers of ribonucleotides. This supports the hypothesis that polymers could be synthesized on the surfaces of different types of materials like sand, rock, clay, and other mineral surfaces that were present on primitive Earth. In addition to forming complex macromolecules, it is also essential to form molecules that can replicate themselves, as living organisms are known to reproduce, and this reproduction requires the replication of their genetic material. The first self-replicating molecule that was thought to be present on primitive Earth is RNA. Thus, it is proposed that there was an RNA world where there were many different types of RNA molecules. We know now that there are some RNA molecules called ribozymes, which are able to catalyze different types of chemical reactions. Thus, it can be perceived that there were ribozymes in this RNA world that had the ability to replicate the RNA. There is more support for the RNA world because when we look at the different molecules present in living systems that have the ability to store and provide energy, they all tend to be ribonucleotides. For example, ATP or adenosine triphosphate is a ribonucleotide that is able to store energy in the form of high energy phosphate bonds. In addition to ATP, NAD or nicotinamide adenine dinucleotide, as well as FAD or flavin adenine dinucleotide, are known to provide and store energy associated with high energy electrons. In order for living cells to form, it is important that these self-replicating molecules as well as other macromolecules are present in a space that is separated from the environment. It has been proposed that in primitive Earth, vesicles were formed, which are fluid-filled compartments enclosed by membrane-like components, similar to the lipid bilayers that we see in today's cells. Scientists have found that vesicles can form spontaneously when amphiphilic molecules are present in water. An amphiphilic molecule is a molecule that has a hydrophilic or polar region as well as a hydrophobic or a non-polar region. Scientists performed an experiment to determine the formation of vesicles in the presence of a type of clay that was known to be present on primitive Earth called Montmorillonite. And what they found was when the amphiphilic molecule or myristoylate was present in the absence of clay, vesicles were formed, but however, it took a long time for them to be formed. However, in the presence of the clay, the scientists were able to see a remarkable increase in the rate of formation of vesicles. Thus, it is a valid assumption that the presence of clay in different parts of Earth allowed the formation of vesicles. We have evidence that macromolecules can form on these Montmorillonite or clay-like surfaces. And thus, when scientists did an experiment where they had RNA present on this clay surface, and when they formed vesicles, they were able to observe that the vesicle, as shown in that circle, was able to take up RNA that's present in that blue circle, and is able to thus demonstrate that we can have macromolecules enclosed into these vesicles. Thus, vesicles with self-replicating molecules like RNA were a perfect recipe to form primitive cells. Thus, as the years went on and all these different developments occurred, ultimately, around 3.5 to 4 billion years ago, the first living cells were formed on Earth. These cells underwent many changes as time passed on to form the different types of organisms that we see today on Earth. With this, we come to the end of our talk, where we learned about the different steps needed to form living cells on Earth. This included the formation of simple organic molecules, followed by the formation of complex organic molecules that are normally found in living systems. We also looked at the formation of self-replicating molecules and ultimately how primitive cells are formed.